Hi guys, today we are going to discuss about de novo synthesis. De novo synthesis is a process of synthesizing fatty acid. And the question arises, why does the body synthesize fatty acids? And the answer is to store energy. I have divided my chart into three parts, that is the pre-requirements and the actual synthesis and post-requirements, that is what happens after the synthesis of fatty acids. Let us start from the first part. Where does the de novo synthesis occur? It occurs in cytoplasm of liver, adipose tissue, kidney, brain, and mammary glands. De novo synthesis is called by many names in other books also. They are linen spiral synthesis, extra mitochondrial synthesis, and fatty acid synthesis. What are the pre-requiring substances for de novo synthesis? That is acetyl-Coenzyme A and NADP. Now, transport of acetyl-Coenzyme A. We got to know that acetyl-Coenzyme A is necessary for de novo synthesis. The acetyl-Coenzyme A is produced inside the mitochondria. That is, inside the mitochondria, it should be moved from the mitochondria to the cytoplasm. Acetyl-Coenzyme A is transported from mitochondria to cytoplasm in the form of citrate. Why is it necessary? Because acetyl-Coenzyme A is impermeable by the inner membrane of the mitochondria. Citrate is transported to cytoplasm by the malate oxalate shuttle pathway. Next comes the sources of NADPH. The two main prerequisites as above is acetyl-Coenzyme A and NADPH. Sources of NADPH are pentose phosphate pathway, active lipogenesis, exose monophosphate shunt pathway, and malic enzyme. Enzymes involved in the de novo synthesis exist as a dimer, as a big complex in which all enzymes are attached. There are three main domains, that is condensing unit, reduction unit, releasing unit. The same three units are present in the lower part also. That is the reason why it is called dimer. The synthase multi-enzyme complex consists of 3 domains and 7 enzymes. First domain or condensing unit, it consists of the following enzymes, that is beta-keto-acyl synthase or condensing enzyme, or acetyl transferase, and malonyl cyclase. The second domain is called the reduction unit. It consists of 4 enzymes, that is dehydratase, enoyl reductase, beta-keto-acyl reductase, and acyl carrier protein. The third domain is releasing unit, which is involved in the release of the fatty acid. In our case, the main fatty acid is palmitate. Now we are going to the actual process of de novo synthesis. The first step in the de novo synthesis is carboxylation of acetyl-Coenzyme A. In this process, acetyl-Coenzyme A is converted to malonyl coenzyme A. Acetyl-Coenzyme is a 2-carbon compound and malonyl coenzyme A is a 3-carbon compound by acetyl carboxylase. In this process, 1 ATP is utilized. The second step of de novo synthesis is divided into 2 parts, 2A and 2B. In the second step, acetyl-Coenzyme A is attached to many acetyl-Coenzyme A to form a long fatty chain. Malonyl coenzyme A is converted to malonyl coenzyme A long fatty chain by the malonyl transcyclase. Malonyl transcyclase transfers the malonyl group to SH group of the ACP of one monomer. The third step is called the condensation step, in which acetyl and malonyl unites and condense to form acetoacetyl ACP. In this process, 1 CO2 is released. The next step is in which acetyl ACP is converted to beta hydroxy butyryl ASP. In this process, 1 NADPH is utilized. The next step is beta hydroxy butyryl ACP is converted to enoyl ACP. That is this is also called as a dehydration step because 1 molecule of H2 is released. Next, enoyl ACP is converted to butyryl ACP. In this process, NADH is utilized. NADPH. Next, the butylate ACP is converted to palmitic acid. This is the final step in which one molecule of H2O is utilized. It is utilized but not released. Palmitic acid, as we know, is a 16 carbon compound. To form this 16 carbon compound, the third, fourth, fifth, sixth steps are repeated in a cyclic reaction for 7 times to form this palmitic acid. Let us see the summary of de novo synthesis, in which 1 acetyl-coenzyme A plus 7 malonyl coenzyme A plus NADPH plus 14H plus gives out 1 palmitate, 7 CO2, 14 NADP plus and 8 coenzyme A and 6 H2O. Regulation of fatty acid synthesis. Regulation of fatty acid synthesis takes place by 4 ways. First one is availability of substrates. As we saw above, the substrates for de novo synthesis is acetyl-coenzyme A and NADPH. As acetyl-coenzyme A is transferred to citrate and taken out of mitochondria to move into cytoplasm, citrate is also an important substrate for the synthesis of fatty acid to take place. Next is acetyl-coenzyme carboxylase. This is a key enzyme in this process because it is used in the very first step of the de novo synthesis. Next comes the insulin. Insulin favors lipogenesis and increases the activity of pyruvate dehydrogenase, acetyl-coenzyme A, and carboxylase. As we know that the insulin increases the glucose level. As a result, the more of glucose is present in blood, which enters the de novo synthesis to produce fatty acids. Glucagon inhibits lipogenesis. Most of the students get confused between beta oxidation and fatty acid synthesis. These are some of the differences between beta oxidation and fatty acid synthesis. The site of action for beta oxidation is mitochondria, whereas for fatty acid synthesis is cytoplasm. Intermediates are present as coenzyme A derivatives and covalently linked SH group of ACP. Enzymes present as independent proteins. In fatty acids, we have seen the multi-enzyme complex. Sequential units are 2 carbon units split of acetyl-coenzyme A and 2 carbon units added to 3 carbon monoyl coenzyme A. Coenzymes are NAD and FAD, whereas for fatty acid synthesis is NADPH. Transporters are keratin, and for fatty acid synthesis it is citrate. Regulation. In our case, if insulin increases, it favors fatty acid synthesis, whereas glucagon inhibits fatty acid synthesis. The most common inherited disorders in case of fatty acid synthesis is dicarboxylic aciduria and Zellweger syndrome, also known as cerebrohepatorenal syndrome. In Zellweger syndrome, it is passed on from parents to children. It disturbs the cellular function and causes serious problems soon after birth. Newborns can have brain, liver, and kidney problems as well as difficulty in feeding or moving. The sad part is that this disease has no cure or treatment. Thank you guys. This is an Ignited Medico signing off.